It can happen, so in a substrate level phosphorylation, for example, there would be a compound, like 1,3-bisphosphoglycerate, going to 3-phosphoglycerate, where here there's 2 phosphates, here there's 1, and the other one ended up on ATP. And this hydrolysis here is highly favorable, and this synthesis is unfavorable, but not by as much. And so this is a coupled reaction by which you can actually make ATP on the surface of this enzyme, which is one of the later steps in glycolysis. Right? So as I said, this kind of thing is getting you maybe 5% of the total energy in the cell. And the other 95% is done by this process of oxidative phosphorylation. And the way oxidative phosphorylation works is that there's been a transfer, is that these energy-rich fuels, like glucose, transfer electrons to redox cofactors, like nicotinamide adenine dinucleotide, which we will see the structure in a second. And then they reduce that cofactor. And then that cofactor interacts with something else and reduces that, which reduces something else, which reduces something else. So the electron's getting passed down and down and down and down. And ultimately, if you're in an aerobic cell, the very last electron acceptor is oxygen. So there's an electron transport chain, which starts with the reactions that we're going to learn on Friday. And the key aspect of the reaction, one of the reactions we'll learn on Friday, is that we're going to synthesize this molecule called NADH, which is the reduced form of this cofactor. And that NADH, being reduced, can reduce something else and become oxidized, and then go back to glycolysis and get another electron, actually a pair of electrons. Right? So oxidative phosphorylation works by an electron transport chain which delivers electrons through redox reactions to the mitochondria, at least in mammalian cells or animal cells, or cells that have mitochondria. And then the energy in these transfers is used. Sorry. So, electron transport chain. Electrons to mitochondria. Build a pH gradient across the inner mitochondrial membrane. Use the pH gradient to drive ATP plus PI goes to ATP. Okay? So that's the process of oxidative phosphorylation. Maybe you got it in outline if you took a cell biology course or something like that. We're going to look at that process in some substantial detail. Okay? And this is on the order of 95% of the ATP in an aerobic cell will be made this way. Okay? So this is kind of like preliminary, so you just kind of get a sense of where we're going and you have some more context for what we're about to do. Yeah?
To like keep homeostasis, right, at a certain temperature, is this related to like the heat that gets released along the way?
There are mechanisms that conserve heat in the process, especially in newborn infants. There are decoupling processes associated with electron transfer that deliberately, apparently, waste heat so that newborns can stay warm. That's pretty neat. But in general, the cell doesn't waste a lot of heat. I mean, to the extent that things warm up, you've lost some of the internal energy of glucose, and you haven't done anything except increase entropy. So, yeah, generally you don't want that. Okay, so we're going to unpack this throughout the rest of the term, right? But this gives you a sense of kind of where we're going. So let's look at some structures for these cofactors. So this is NAD. I don't expect you to be able to, you don't have to be able to draw it from scratch. You have to be able to recognize it and understand how it works. And we will see some aspect of the mechanism soon. But the structure of NAD, this is the oxidized form. And notice that it's got an adenine ring, a ribose type sugar, right? Whenever we find these cofactors like ATP and NAD, and they always have sugars in them. The sugars are always ribose, kind of like reflecting the RNA origin of life kind of deal. At least that's one of the pieces of evidence that gets pointed to. There are two kinds of nicotinamide cofactors, NAD and there's NADP. And the difference between them has nothing to do with the chemistry of the redox part. It's just that NADP has this extra phosphate group, which helps basically bind it to the enzyme. Okay. However, having said that, it's also true that when we look at the picture of metabolism more broadly, NAD will get used in certain metabolic contexts, and NADP will be used in other ones. And so the cell is very careful about keeping them apart. Even though as far as their redox function goes, it's the same. Right? So that phosphate is down here when it's present. And then it would be just NADP plus. Right? The plus here, of course, you've got negative charges on there, so you could say, well, I see a plus there, but two negatives, so it should be NAD minus. But by convention, that plus refers to what's going on in the nicotinamide ring only. So we kind of forget this is like so much cabbage going on out there. Although, of course, it's important for any enzyme that binds it, it's going to have binding determinants on the cabbage. So this is the oxidized form of the nicotinamide ring. So you can see what the structure of that ring looks like. It's a heterocycle with one nitrogen in it. Right? It's got this amide coming off one position of the ring. And it accepts 2 electrons and 2 protons to form an uncharged structure that also has an additional hydrogen on here. And a proton is released in that mechanism. So we'll see how that works in the context of a particular enzyme, which is a dehydrogenase. So the class of enzymes called dehydrogenases are enzymes that carry out redox reactions, i.e. we had 6 categories of reactions, or 6 categories of redox. Oxidation reduction was one of those categories. Dehydrogenases are a common enzyme in that redox category, and they use NAD or NADP. Right? And they convert it to NADH. Once they've converted it, you should ask, this is a generalized reaction here. Where these protons could come from another substrate. They could get picked up from solvent, for example. Right? Depends on the mechanism. Where are these electrons coming from? Right? So something is delivering electrons to this. Something else that is reduced is delivering electrons to this. Right? And in the context of glycolysis, of course, what's reduced is the sugar. Right? So it's not going to be glucose directly, but somewhere along the line, electrons are coming out of the sugars and getting into NAD plus to form NADH, which is then going to go on and do its thing in the mitochondria. Right. This is just showing you that there's a different absorbance spectrum for NAD plus versus NADH, and that means that it's easy to detect them in enzyme reactions. So if you got a dehydrogenase and you want to assay it and you have the right substrates, add the enzyme, close your spec, hit return. The data accumulates in your computer as the reaction goes on. Okay? If you're forming NADH, the absorbance at 340 is increasing, and all you have to do is track it, and next thing you know, you've got kCal. So these are the important facts about NAD that you should know. It's soluble and diffusible in cells. That's very, very important because some cofactors aren't, and that controls or shapes how metabolism is set up. Okay. The substrate is oxidized. The substrate meaning whatever the other compound is in the reaction. So NAD is, if you can think of it as a substrate, you can also think of it as a cofactor or both. It doesn't much matter if you call it a substrate or you call it a cofactor. But there will be some other molecule in there, like some sugar compound. And that sugar compound will be getting oxidized according to the rules we learned on the first slide. Right? so that this guy can get reduced. And that'll be one of the steps along some metabolic pathway. Right? So the substrate is oxidized, and hydrogen atoms are also removed from it. If not two, then maybe only one, and one hydrogen comes from solvent, something like that. But often 2 hydrogens are removed from substrate, and 2 electrons are removed from substrate. 2 electrons and 1 proton are transferred to NAD, the oxidized form, to reduce it. And the way that they're transferred is as a hydride ion. So remember, that was one of the ways in which we could transfer electrons. Okay. The remaining proton is released to solvent. There can also be, in some cases, some stereospecificity of hydride attachment to the ring. So this ring, if you look at it from the front or you look at it from the back, it's a little subtle. And this is not something that I really want to dwell on, but the textbook goes into it a bit. Sometimes these dehydrogenases will, in the details of the mechanism, come from one face of this ring or come from the other in terms of adding electrons to it and adding proton to it. Okay, so those are the important facts about NAD. And I mentioned, or most of them, I mentioned that NAD and NADP have different roles in cells. In oxidations, like what we're about to learn for glycolysis, it's NAD that's more used. In anabolic reductions, meaning biosyntheses, right? Anabolism is biosynthesis. Reduction means we're making something, we're reducing it, right? Those reactions tend to use an NADPH. So we'll see examples of that. Burning fuels and synthesizing new fuels, right? Or synthesizing large components to incorporate into the cell if they're not going to be fuels.